This is the light reaction. Light reaction actually involves these four processes. These are the four processes: photoexcitation of chlorophyll, photophosphorylation, which may be cyclic or non-cyclic, production of NADPH, the reducing potential, and photolysis of water with release of oxygen. So these are not for different steps. These are the things involved, or the purpose of light reaction is shown here. So this is photoexcitation of chlorophyll. This is already I described. The photoexcitation actually happens with the fall of photons on leaves. Photophosphorylation means using the photon and the energy of photons. Phosphorylation or production of ATP is used is done, and it can be through two different types of cycles, types of processes. One is cyclic photophosphorylation, the other one is non-cyclic photophosphorylation. Then production of NADPH, so that is important, reducing potential, as well as release of oxygen, that is another one, that also is very important. So this is called Hill reaction after Robin Hill. Photoexcitation of chlorophyll, absorption of photons by antenna molecules, electron gets excited. This is already discussed. We already discussed in the previous slides. Photoexcitation of chlorophyll means absorption of photons by antenna molecules. So antenna molecules in the cluster, the photosystems 1 and 2, these molecules, the particular molecules, accept photons from solar energy. As a result, their electrons gets excited. The excited electrons transfer their excitation energy to the, it is actually a short-living process, and it will transfer to the adjacent molecules. And this process is called sensitized fluorescence. As a result, the first one which transfers energy, the electron will come to ground energy level. And finally, the excitation passes through certain molecules and finally reaches the reaction center or the principal chlorophyll A molecule. Okay, after photoexcitation of chlorophyll, we move on to see cyclic and non-cyclic photophosphorylation. This is actually the excited electron released from the principal pigment travels through certain this high energy electron with a high reducing potential travels through certain protein cluster assemblies. In cyclic photophosphorylation, the electron which left the principal pigment reaches the same point, whereas in non-cyclic photophosphorylation, the electron do not reach the same point from where it was left. It is going through some other, we will see some other pathways. It takes some other pathways. The purpose of these photophosphorylation reactions are that the cyclic photophosphorylation results in production of ATP, whereas non-cyclic photophosphorylation results in the production of ATP, NADPH, and it causes photolysis of water, or photolysis of water is a process which takes place during non-cyclic photophosphorylation along with release of oxygen from the water molecule which is split. Now, one of the basic ideas of cyclic and non-cyclic photophosphorylation where it actually results in ATP synthesis is that you can see here the mesophyll cell membrane, this is the mesophyll cell membrane, this is mesophyll cytosol, and inside which chloroplast outer membrane, inner membrane, stroma, and the thylakoid. Thylakoid is here with the lumen, as you can see this lumen. So here what cyclic and non-cyclic photophosphorylation do is that these are specifically as far as ATP synthesis is concerned, they specifically lead to accumulation of H plus ions. This hydrogen plus, as you can see, the accumulation of H plus ions inside this thylakoid lumen. So this is a process which will lead to ATP synthesis later. That we will discuss later. But ATP, whether it is cyclic or non-cyclic photophosphorylation, it leads to accumulation of H plus ions here. So as a result, what happens is that there will be an electrochemical gradient between thylakoid lumen and stroma. thylakoid, lumen, and stroma, electrochemical gradient. This is because of the accumulation of H plus ions inside this lumen. Yeah, electrochemical gradient. And this is actually a proton gradient, an electrochemical gradient that will be the energy which is responsible for ATP synthesis. Now, first of all, we can go through cyclic photophosphorylation. As you can see, the photo excitation of chlorophyll is shown here. The energy of photons, high energy electron is released from here. It travels through certain clusters and reach back to the original position. This is why it is called cyclic photophosphorylation. Now, it involves PES1 only, photosystem 1 only. Obviously, chlorophyll A700 is at the principal pigment or the principal position. Now, the process generates ATP only and not NADPH, and there is no hydrolysis of water. As a result, there is no, consequently, there is no release of oxygen also. Now, we can study cyclic photophosphorylation here using this diagram. As you can see, this is a PS700 from where upon light fall of photons, the high energy electron is released from PS700. The high energy electron moves to a highest level like this. That is actually along a redox potential. It is uphill of a redox potential. It reaches here, where it will be accepted by a primary electron acceptor. From there, it goes to like this. It goes through certain the electron acceptor assemblies and come back to the same position. But as it goes through, first of all, it reaches the ferredoxin. It's a iron ferrous sulfur protein cluster, from where it reaches cytochrome BF complex. It's actually a proton pump. It is capable of accepting H plus while it accepts electron from the ferredoxin molecule. It accepts H plus ions from stroma, and as it releases the electron towards or to plastocyanin here, plastocyanin is a copper containing protein assembly. As it loses electron, it actually releases the proton to the thylakoid lumen. So protons are taken from stroma and released to thylakoid lumen. So as a result, thylakoid lumen gets accumulated, gets concentrated with H plus ion, this proton. So this is what I have shown in the previous slide, H plus ion concentration or accumulation inside thylakoid lumen because of cyclic photophosphorylation. Now, from plastocyanin, the electron it travels down the downhill of the redox potential here to reach back to PS700. Now we can see the process here, actual process of H plus ion concentration, accumulation. Now this is stroma, lumen, cytochrome BF complex, electron passes to cytochrome BF, and what happens is that it accepts H plus from stroma, and as the electron leaves cytochrome BF complex, H plus ion is released, like here, released to lumen, then causing accumulation. So this process, as the process repeats, H plus ions are further released into lumen. Now this is non-cyclic photophosphorylation. As we know, it actually causes ATP synthesis. It results in NADPH production as well as photolysis with the release of oxygen also take place in non-cyclic photophosphorylation. Now this involves photosystem 1 and photosystem 2, both of them. And this is how photosystem second is the first one involved. This photon, a high energy photon comes, high energy electrons are released from principal pigment of photosystem second, that is 680. It travels downwards, like this, down the downhill, the redox potential, and it do not reach back to PS680 but falls on the next photosystem 1, and from where a high energy electron from P700 is released Yeah, like this. This is caused by reception of further photons on photosystem second. As you can see, light energy, the sunlight falls on this photosystem 1, electron is released, and this electron is accepted by a primary electron acceptor. And mostly these primary electron acceptor is a pheophytin, a molecule called pheophytin, from where it travels further towards reduction of NADP plus. This is a better illustration of the process of non-cyclic photophosphorylation. And the photosystem second with the P680 at its center, the principal pigment, upon which photons fall and electrons expelled from the principal pigment. High energy electron is accepted by primary electron acceptor. Mostly it is pheophytin. From that highest level, it travels down the energy level or downhill the redox potential. First of all, passes or accepted by plastoquinone, which gets reduced to plastoquinol upon acceptance of a pair of electrons. Then those electrons pass through cytochrome BF complex, which is actually a proton pump. It accepts protons from stroma, and that is released to the luminal space. So this is a process which contributes to the synthesis of ATP later. Now this electrons passes through plastocyanin, and finally it is accepted by the photosystem first here. And now photons fall on it, on this photosystem first, and finally the P700 principal pigment releases a high energy electron here, and that electron or those electrons are actually accepted by primary electron acceptor, then goes down the energy level, accepted by ferredoxin here, and then a couple of electrons travel to reduce NADP plus to NADPH. So this is how NADPH is formed. Now the NADPH is actually formed with the help of a couple of protons also taken from stroma, like here. It is actually taken from stroma, and this is actually catalyzed by ferredoxin NADP plus reductase enzyme. Now at the initial point here on the photosystem second, there happens what is called photolysis of water, that is splitting of water with the help of photons and with the help of a particular protein, enzyme like protein, a protein which is a manganese protein because it contains manganese molecules in it, 4 of them per protein molecule. And now this manganese is evolutionarily selected for this purpose. It is said that it's because of the capacity of this manganese to exist in different oxidation states like Mn2 plus, 3 plus, 4 plus, and Mn5 plus. Now, as a result of photolysis, what happens is that oxygen is released. Per 2 molecules of water split, a molecule of oxygen is released, as well as the electrons. 4 electrons are released, which actually fill the hole created at P680 here. So you can assume that these electrons through this, through this electrons passes, electrons travels, it is those electrons which actually reduces NADP plus to NADPH also. Now what remains is H plus. Those H plus, 4 H plus molecules are again they are released to thylakoid lumen. So this again contributes to the synthesis of ATP later. And further, one more point there is that this is called Z scheme also. It is just because the energy level shown here, like this is how the electron travels. It's like the English alphabet letter Z. So this is called Z scheme. Now 2 types of photosystems cooperate. So this is what we have already discussed. Photons here, photons here. This is ATP mill, cytochrome BF complex, and NADPH production. And this also we have discussed, the production of oxygen, that is from water, not from carbon dioxide. This has been proved by radioisotope studies. Actually 2 molecules of H2O are oxidized to generate a molecule of oxygen for every 4 electrons passes through this transport chain. This is the equation for it. and we have already discussed all these things. And this is a representation for accumulation of H plus inside thylakoid lumen, or inside thylakoid luminal space. Now this is the membrane which differentiates stroma from thylakoid space, and it is actually inside the thylakoid space that H plus accumulates. It is through these processes. This is photolysis, H plus ions accumulate here, or after photolysis whatever H plus is released to the interior of thylakoid space. So accumulation of H plus. Here again, cytochrome BF complex is a proton pump that also help accumulate thylakoid space H plus. And from here, what happens is that H plus is taken away from stroma. So this also contributes to the H plus ion gradient here across this particular membrane.